It is no surprise and no secret to anyone that we are very passionate about MRD. Now, traditionally, we have looked at MRD as a biomarker of treatment efficacy and eventually one of the most relevant, if not the most relevant prognostic factor in myeloma. Now, there is another side to the story. And the other side is that if we believe that persistence of MRD is one of the greatest barriers towards prolonging patient survival, then we must study the biology of the MRD clone. And this is what we have been doing in the past 5-6 years. Whenever we detect persistent MRD in patients with myeloma treated in the Spanish clinical trials, we isolate the MRD clone and we compare the biology of the MRD clone with tumor cells from the same patient at diagnosis and in some also at the time of relapse. And by performing whole exome and RNA sequencing, we have shown at ASH that the MRD clone is a unique clone throughout genomic evolution from diagnosis to MRD to relapse. And this data could potentially inform us on new avenues, new pathways to target MRD resistance, and can also help us to better understand some clinical aspects of treatment, such as, for example, the fact that in many patients, continuing treatment with the same drugs is not able to overcome MRD resistance. And the reason may be because the MRD clone is completely different from the clone that was predominant at the time of diagnosis.